Swayam Prabha. Digital India. Educated India.
Welcome back to my course Industrial Biotechnology. In the last class I tried to point out that how the biological treatment processes, wastewater treatment processes plays important role in the industry. I told you whatever organic waste discharged by the different chemical and biochemical industry, more than 70% of that is treated by biological means. And we have two processes with us. One is aerobic wastewater treatment process, another is anaerobic wastewater treatment process. In the last two lectures I tried to cover the activated sludge process, and also will deal with the wastewater characteristics, and I made a comparative study that how the carbon and energy balance that we have in the both aerobic and anaerobic wastewater treatment processes. Now, one thing what I discussed in the last class, I want to little bit clarify that FM ratio, that is food microorganism ratio, when you design the activated sludge process is like this, that F by M is like this, and this can be written as I told you food is substrate, that is F0 into X0 and divided by V into X. Now, F is the flow rate, volumetric flow rate, you can see this is the volumetric flow rate, and the unit is volume per unit time, and the X0 is the concentration is of this concentration of substrate. This is is the mass per unit volume. So, volume will cancel, so it will mass per unit time, and here this is the volume of the reactor and this is the concentration, this is mass per unit volume. So, if this will cancel, so what we will get, this is mass will cancel, so the unit will be time inverse. Now, here is I told you this is equal to S0 V by F0 into X. Now, V by X0 is the hydraulic retention time, so it is theta into X. This is how in the last class I did not clarify that. So, I hope this should be clarified. So, this will be this S0 by X into S. Now, today my topic is anaerobic treatment processes. Now, before I begin, let me tell you the advantages of this process. Now, I told you that whenever we go for installation of any chemical and biochemical industry, that we shall have to show that that kind of effluent that is generating by the industry, that should be properly treated, because we have no right to damage the environment. So, Central Pollution Control Board, they put some kind of rules and regulation just to safeguard our environment. They have disposal standard, and that every industry should follow. Now, when any industry go for that in the early occasions, that they found that 10% of the total expenditure of the industry goes for the wastewater treatment process, but in a true sense the activated sludge process does not give any monetary feedback from the process, because most of the money that you spent, that you cannot get much of return from that. But due to the development of this anaerobic digestion process, now it is possible that some of the money that you spend for the wastewater treatment process you can get back. I can give a typical example, the IIT agro industry that is located close to Calcutta, that and they are using the anaerobic digestion process for treating their distillery effluent, and they observe that 50% of their distillery and distillation plant energy required for the distillation plant can be can be shared from this methane and methane that produced from the anaerobic digester. So, it is a great avenue, and not only that, I shall show you that different western country and also they are largely using this anaerobic digestion process and use for different purpose. I can give the example that if you go, I have seen in Sweden, I have seen in Germany, they are running the buses with the help of biogas, and also in India also now we started using biogas for running the buses. So, it has great different uses that we have. So, and not only that, if you look at the history of biomethanation process, what we call biogas generation process, it is very old. It is invented by American scientists or Western scientists, and since our the economic conditions of our country as compared to the Western country is a little bit poor as compared to Western country. So, it is largely applied because this technology in both in India and China, because this has both small scale as well as large scale applicability. And India, we have millions of biogas plants that is located in the villages, and also China, they have the millions of biogas plant in the villages. So, you know that they take the advantage of the degradation of this raw materials, organic materials, and generate the methane and carbon dioxide, and that is used for cooking purpose or some other purpose that and digested material they can use as a biofertilizer. I told you this biofertilizer is very good because this increase the water retention property of the soil. This is how the fertility of the soil increases. Now, let me start this. This is if you look at methane, that is gas that is produced through this anaerobic digestion process, is a colorless, odorless gas. It does not have any kind of odor, and primary component of the natural gas, because if you look, because I can tell you, I can share with you some information that in a couple of years back, that in Delhi we have lot of environmental pollution problem due to use of petrol and diesel, and because lot of particulate matter, lot of the fog has occurred. So, Government of India, they implement the CNG, compressed natural gas, for running the vehicles, and by using this fog problem has been reduced drastically. So, this is used from the natural gas, and so it is the primary component. Methane is the primary component of the natural gas. Chemical formula is CH4. Now, methane is the chief component of the natural gas, is produced in nature by bacterial decay of vegetation and animal waste, because I shall show you how the degradation take place. And this process is called anaerobic digestion. Now, anaerobic digestion is a series of biological processes in which the microorganism break down the biodegradable organic matter in absence of oxygen. One of the end products of the biogas is the biogas, which is combusted to generate electricity and heat, or can be processed into renewable natural gas or and transportation fuel, as I mentioned before. So, this is the organic waste. Then the organic precipitation, we preparation, then we pass through the anaerobic digestion process, we get the biogas. So, then it can be used, heat and electricity generation we can use, and digested material can be used as an organic fertilizer, and this water you can recycle back to the system. So, this is the process largely applicable throughout the world. And if you look the steps of that degradation process that is involved in this anaerobic digestion process is like this: hydrolysis. First, the organic material, carbohydrate present in the form of starch, cellulose, and then we have proteins, we have fats, all are bigger molecule. They hydrolyze. I told you hydrolysis takes place when any water molecule involved in this reaction. It produces sugar, amino acid, and fatty acid, and it passes through the acidogenesis, where through which they produce volatile fatty acids, and this volatile fatty acid again converted to acetic acid and hydrogen and carbon dioxide, and through the methanogenesis it will converted to methane and carbon dioxide. So, here I want to point out that two groups of microflora that take part in this system. This hydrolysis and the acid production is usually take place by the acidogens, and methane production taking place by the methanogens. So, two group of microflora, because they are totally different from each other, they are not same. So, acidogens, they convert organic waste to volatile fatty acid, and methanogens they convert this to methane and carbon dioxide. Now, if you look at the characteristics of the acidogen, characteristics of the methanogens is totally different. So, but I can tell you you have seen here the substrate required for acidogens is organic waste, which comprises of different polymeric compounds, different bigger molecules, like you have cellulose, hemicellulose, starch like this, also protein, fats like this, that are hydrolyzed. I showed you here, and finally it produces volatile fatty acids like acetic acid, butyric acid, propionic acid, like this. And these acids they utilized by the methanogens to convert it to methane and carbon dioxide. So, the characteristics those two is different with respect to substrate they use. Not only that, with respect to pH, that pH, these acidogens work at the acidic pH, maybe 5 to 6 is better for this, and in case of here it is 7.5 easily better, that is the alkaline pH is better for the growth of methanogens. Now, if you look at the growth of the acidogens is quite faster, this is quite faster, and this is slower. So, this is slower. So you know that I showed you before that when we did the reactor analysis, then I showed you that how the dilution rate that that the affects that what do you call x into d. X into d is nothing but dx by dt, rate of growth of the cells. It is like this. So, this is for the slow growing organism, and this is for the fast growing organism. So, you can see this is the, I told you this is the D washout, and this is the D washout. This is one washout. So, if you look at that that faster required more, the D washout value is much higher as compared to slow. So, if we maintain the dilution rate here, so we can easily wash out the methanogens from the culture. So, you can do it very easily. So, this is we can produce the acidogenic culture separately and methanogenic culture. Methanogens we can develop by taking the mixed culture and put the selective substrate. We put only the volatile fatty acid as a substrate in the reactor, so that only methanogens can grow, because methanogens can only convert the volatile fatty acid to methane and carbon. So, we can segregate the culture like this. So, that raw materials used for this particular process is animal dung, that is like cow dung, lot of animal dung so we have. Then poultry waste, I discussed that how one particular family in some city, that how he be able to run his poultry farm without by taking care the public opposition, because anaerobic digestion remove that all the bad smell that is generated by this particular waste, because this is a closed chamber. This is anaerobic digestion, means it doesn't require any kind of oxygen, so no contact with the air, so no gas, and digested material doesn't have any smell, so it will be totally free from the odor. Then plant waste like hogs, grass and weeds, etc., human excreta, industrial waste, sawdust, waste from the food processing industry, and the domestic waste like vegetable peels and waste food materials. So, these are the different raw materials can be used, and if you pictorially it can be explained like this. We have energy crops, manure, industrial waste, and different restaurant waste and wastewater. When passed through the anaerobic digester, it looks like this, and it can be used as a renewable fuel. For digested material can be used as a biofertilizer, which increases the growth of the plants very high. We can use as a for the generation of electricity. Now, if you look at how the degradation take place, I told you this is lipid, polysaccharides, proteins, and nucleic acid. The lipid will degrade it to fatty acid, polysaccharide will degrade to monosaccharide, proteins will degrade it to amino acids, and nucleic acid will degrade to purine and pyrimidines and simple aromatics. And this again converted to the volatile fatty acids like propionic, butyric, and lactic acid, and this ethanol, and also acetic acid. But this is higher acid. This passes through the acidogenic process and convert the acetate higher organic acid like butyrate and propionate. When it converted to methane and carbon dioxide, first they produce the acetate, what we call acetogenesis, and then this acetate is converted to methane and carbon dioxide, and also hydrogen and carbon dioxide they combine to produce the methane. This is how they place. So, I was talking about this is methanogens, and this is the acidogens, how they take care of the reactions. Now, if you look at this how the reaction take place, this is another way we can explain. This is similar, but in a different way it can be explained like this. And Barker hypothesis for the biomethanation methane formation, he has explained like this how he take care. In this particular pathway, there is a coenzyme called coenzyme N that very much take part in the reaction, and that is how is carry forward and produce methane here. Again this is carboxylation and hydrogenation, then again reduction, again it is further reduction to methyl groups, then it produces methane, and this is also can be produced, this can be produced with methyl alcohol and acetate like this. This is the Barker hypothesis, and this is the thermodynamics behind this process. And when the glucose is converted to acetic acid, your free energy change is minus 74.3 kilocals. But when this carbon dioxide and hydrogen produces acetic acid, this is very low, and carbon dioxide produces methane, that is also comparatively low. So, in this reaction I can always say that glucose to acetic acid is more favorable as per this reaction is concerned. Then after that this methane production, after that this acetic acid from carbon dioxide and hydrogen. Now, another thing very interesting, the when we considered acetic acid, when it converted to methane, the free energy change is minus 7.4 kilocals, where your propionic acid you required plus say 19.5, and butyric acid is 9.95. From this we can easily say that propionic acid degradation is very much less favorable as compared to that of acetic acid and butyric acid. So, it has been observed when you operate any kind of anaerobic digester for a longer period of time, there will be accumulation of propionic acid in the reactor. Long run, as you operate for longer period of time, the propionic acid accumulation in the reactor because the propionic acid degradation will be very poor. Now, this is the Busoil equation, how the stoichiometry equation, how methane can be calculated, and Klaas and et al. They also did lot of work on this biomethanation process. They use this equation. This is the empirical formula of the kelp, is kind of seaweed, and this when it converted to methane and carbon dioxide, the stoichiometry is like this. Now, there is a scientist called Sanderson et al. Reported view disproportion reaction, because this is very interesting that I told you in the anaerobic digestion, the acidogenesis process, we get the volatile fatty acid, and volatile fatty acid mainly comprises of acetic acid, propionic acid, and butyric acid. Now, this is the dispropionation reaction, that is that means usually we express the concentration of volatile fatty acids in terms of acetic acid. If you want to express like this, 5 moles of acetic acid is equivalent to 2 moles of butyric acid, 7 moles of acetic acid is equivalent to 4 moles of propionic acid. This is the disproportionate equation developed by Sanderson. Now, microbiology behind this biomethanation process, the methanogens belong to the Aequiobacter. This is kind of special type of seed. They are distinguished from the Eubacter bacteria by virtue of many construction characteristics, such as the presence of isoprenoid rich membrane lipids. So they are typical characteristics that as compared to and they link with the glycerol, absence of mauramic acid based on peptidoglycan cell wall and distinct ribosomal RNA. So, this is we have 3 type of methanogens. One is carbon dioxide reducing, another methylotrophs, another is acetoclastic this pathway. So, carbon dioxide reducing, carbon dioxide is converted to methane, methanotropic that we also here also is converted, carbon dioxide converted to methane by oxidizing methyl group containing the substrate such as the methanol, trimethylamine and dimethyl sulfate. Acetoclastic usually convert the acetate to methane. These are the different methanogens present. One is methanobacterials, then methanococcals, then methanomicrobials, methanosarcianals and methanopyrals. So, the different type of methanogens, they are all obligatory anaerobe, and this is how they take part in the reaction. This is shown here, how they use the different substrate for the production of methane and carbon dioxide. Then this is the biochemistry behind that. Biochemistry quality of the methanogenic bacteria are different from other bacteria. A very restricted range of oxidizable substrates coupled to the biosynthesis of methane, synthesis of an unusual range of the cell wall component, the synthesis of biphenyl glycerol ethers as well as high amount of sequelin. This is the synthesis of unusual coenzyme M. I told you this is the typical coenzyme M that present in the methanogens, and some growth factor also required. So, so I was talking about that acidogens and methanogens, and since the acidogens require different growth characteristics as compared to methanogens, it is advisable that if we can operate this process in 2 different stages, then this will be very easy to control, because acidogens they required different environmental conditions as compared to methanogens. Now, if you look at normal anaerobic digester process, they have one particular chamber inside both the acidogens and methanogens, their activity taking place. The rate of there you have to synchronize the process in such a way the rate of acid formation should be equal to rate of methane formation. Otherwise the pH of the system will go down and affect the methane production process to a great extent. This is to be taken into account when you operate any kind of anaerobic digester or the biogas plant, because in our rural places some of the biogas plant doesn't work because the villages they are not aware that how to feed this material to the reactor. Because I can give a very typical example that that I told you that organisms they are very sensitive to the environment, and particularly temperature plays very important role for the activity of the particular organism. So, during winter our ambient temperature go as low as 20-25 degrees centigrade. So, that time your actual temperature for the growth of organism is 35 to 37 degrees centigrade. so activity of the organism will drastically reduce. So then if we put the substrate at the same rate as we have in the hot season, when the temperature, ambient temperature is 40-45 degrees centigrade, then the organism will not function in a similar way. So you have feed rate, you have to reduce to a great extent, so that rate of acid formation and rate of methane formation can be synchronized properly. So this is easily done by using a two-stage reactor. We pass the substrate in the acid regions, and then here the acid formation take place. This acid we take it to the methanogenic reactor to get the methane formation. Now, since the rate of reaction of acid region is much faster as compared to methanogenic reactors, the size of the methanogenic reactor will be much higher as compared to that of acidogenic reactor. Now, this is the kind of analysis, because I personally done some research work on that. I tried to find out that how cow dung is the traditional substrate for the biomethanation process, and how this can be comparable with that of mixed residue, because we have observed that in the any kind of biochemical industry, when it depends on particular raw materials, even that raw materials is cheaper, but after some time you will find that the unavailability of these raw materials. When the people will find that some material has some importance, they restrict for selling that material. So, or they will increase the price of the material. So if you have number of different type of raw materials you can use for that particular process, then the cost of the raw materials can be controlled to some extent. Also, availability of the raw materials, that also increases to a great extent. So we use the mixed substrate concept, mixed biomass. We use cow dung, water hyacinth and algae, and we find this is much suitable. It is not only, if you compare with this batch process, you will find that methane production here 0.24 to 0.246 cubic meter per kg volatile solid digested, and where two-stage process is 0.475. It is double as compared to that, that here it is this. If you compare this two-stage process for cow dung and mixed residue, it is almost double. So this is the energy analysis of this process. We have mixed residue here. This is the total composition. We find during the degradation process, lignin content increases. Here it is 11.7, here it is 16.8, because lignin doesn't take part in the reaction, but cellulose it was 22.1, here 19.8, but hemicellulose is 20.8, here you have 6.8. So what I pointed out, hemicellulose is a very good raw materials for the biomethanation process, which convert the organic material to methane and carbon dioxide. So this is some kind of information that we have. How many biogas plant, that is, this is the year 2013 in the European country, how many biogas plant are in operation. Total is 208, and Germany playing the major role, 107, and then Sweden. I told you Germany and Sweden, they are largely operating their bus with the help of biogas, which is environmentally friendly. So what I try to point out in this particular presentation, that I told you biomethanation is a process through which we find some kind of revenue of the money that you spend for operating that process. So by selling the gas in the form of methane and carbon dioxide, and also digested material also we can sell in the form of biofertilizer, which has also some selling value. So with and this can be easily controlled with the help of two group of microflora. One is acidogens, another is methanogens, and if we control this organism properly, we can run the plant very comfortably, and we should not have any kind of problem for operation. And because if we give this information to our villagers who has operating the biogas plant in the rural areas. They can operate the biogas plant very successfully and make use for their cooking purpose, because it can be used for different purpose. As I show you afterwards, then the biogas has different utilities. So with this, let me stop here, and the next class, lecture, I shall concentrate on how this process is largely applied to the bigger scale, and also I shall give you the information how this can be comparable with the chemical process. And finally, I want to discuss one successful project on anaerobic digestion, what we call landfill gas generation process that is in operation in the Western country. Thank you.